Dear students, welcome. My topic today is seed dormancy and seed germination. In this topic, we'll discuss seed dormancy, types of seed dormancy, seed germination, and then types of seed germination. Seed dormancy first. Angiosperms are the most diverse group of plants. The defining feature of this group is the presence of seed. Seed is considered as a wonder of nature. Seed, in a small package, has got everything of a mature, full-fledged plant, including roots, shoot, leaves, stored food material, and protective coverings. Seed is a special and highly adaptive structure. It's critical in the continuation of a flowering plant's evolution of seed habit has been a key factor in migration of plants from aquatic to truly terrestrial habitats. Seed ensures the survival of plant through unfavorable and hostile environmental conditions. It perinates in the form of an inactive dormant propagule, suspending all activities of life temporarily during unfavorable times. This period of inactivity in the life of a seed, when it fails to respond to favorable environmental conditions, is known as seed dormancy. Dormancy is the inability of a seed to germinate under conditions that would normally favor its growth. Dormancy is very important as it allows a dispersal of seeds and avoids precarious germination. The stalling of germination also safeguards seeds and seedlings from suffering damage or death, which may result due to short periods of favorable weather followed by long periods of inclement and unfavorable weather. Apart from this, seed dormancy also has economic and ecological significance. Dormancy also protects seeds from transient herbivores. Dormancy comes as a pause in life cycle. The level at which dormancy is introduced in the seed has been seen to be species specific and is initiated during the development of seeds. Nikoleva divides seed dormancy into types which include physiological dormancy, morphological dormancy, morphophysiological dormancy, physical dormancy, physical plus physiological dormancy, chemical dormancy, and mechanical dormancy. Number first, the physiological dormancy. When the emergence of a radicle from seed is inhibited by physiological mechanisms of embryo, the dormancy is said to be physiological. Most of the seeds exhibiting physiological dormancy are permeable to water. However, there are some exceptions too. The physiological dormancy has three types. Number first is non-deep physiological dormancy. Seeds with non-deep physiological dormancy include seeds of weeds, many vegetable seeds, and seeds of some garden flowers and that of some woody plants. Seeds with non-deep physiological dormancy after their maturation either cannot germinate at any temperature or they germinate over a very narrow range of temperatures. Number second subtype is the intermediate physiological dormancy. Intermediate physiological dormancy is present in the seeds of Acer nigundo, Acer pseudoplatanus, Acer saccharum, Corylus avellana, Fagus sylvatica, Ferula caratevica, etc. Like in non-deep physiological dormancy embryos, embryos excised from intermediate physiological dormant seeds germinate and the resulting seedlings are normal. Cold stratification breaks dormancy but may take as long as 6 months, depending upon the species. Dry storage at room temperature reduces the cold stratification period in some species, including Amelanchier canadensis and Echinocystis lobata. Number third subtype is the deep physiological dormancy. Species exhibiting deep physiological dormancy include Impatiens parviflora, Malus domestica, Sorbus ocuparia, Rhodotypos caricoides, and Prunus persica, etc. Embryos, if excised from deep physiological dormant seeds, either do not grow or produce abnormal seedlings on germination. Such seeds require very long periods of cold stratification to overcome dormancy, ranging from 7 weeks in Acer platanoides to 14 weeks in Prunus persica. Rosaceous seeds with deep physiological dormancy, if not subjected to cold stratification, produce slow growing abnormal plants. These plants have short internodes, small malformed leaves, sometimes occurring in a rosette at the tip of the shoot called as a perched rosette. The non-cold stratified seeds are thus used to produce dwarf plants, and this is called as a nanism. The dwarf plants in such cases never revert to normal. Number second type of the seed dormancy is a morphological dormancy. The embryo in seeds with morphological dormancy at the time of their dispersal is either differentiated into radicle and pilumule but underdeveloped, or is not differentiated and is just a mass of cells. In both these cases, germination is postponed due to the morphological characters of the embryo. Hence, this type of dormancy is known as morphological dormancy. Now, the first case is that of differentiated embryos. Morphological dormancy has been reported in seeds with rudimentary and linear embryos. Embryo in such seeds may represent just 0.1% or less of the total volume of the seed. Rudimentary and linear embryos are differentiated, that is, the radicle and the pilumule are present in the embryo, but they are small and required to grow before germination could take place. Many plant families contain embryos underdeveloped at the time of dispersal. Growth in underdeveloped embryos takes place only after they have been dispersed. The environmental factors which are important for growth in such embryos include moist habitat, suitable temperature, with some species requiring specific light and dark treatments. The temperature range between 15 to 30 degrees Celsius has been found to be favorable for most of the species with morphological seed dormancy. However, some species like Elais genuensis require temperature between 35 to 40 degrees Celsius, while seeds of Apium graveolens require light to germinate. Those of Conium maculatum germinate to higher percentage in light than in darkness. Number second case is that of undifferentiated embryos. One or a few genera in a number of plant families have embryos undifferentiated at the time of dispersal. Such type of embryos occur mostly in micro and dwarf seeds. Microseeds are usually less than 0.2 millimeters in length and dwarf seeds between 0.3 to 2.0 millimeters in length. Embryos of microseeds can have from 25 to 100 cells, but they may have as few as 2 only. Seeds with undifferentiated embryos do not develop a radicle or cotyledons. Instead, either the radicular or the pilumular pole of the embryo axis elongates and emerges from the seed and forms a hostorium or a protocorm depending upon the species. Next type of seed dormancy is morphophysiological dormancy. This kind of dormancy as a morphological dormancy is found in seeds with linear or rudimentary embryos. However, Underdeveloped embryos here also have physiological reasons for being dormant. Morphophysiological dormancy is found in many plant families including the Apiaceae, Equifoliaceae, Aristolochiaceae, Berberidaceae and Fumariaceae, etc. Seeds with morphophysiological dormancy require overcoming two hurdles before germination in them can begin. Number first, the embryo must grow to the critical size, which is species specific, and number second, it must overcome physiological dormancy in it. Different treatments are responsible for breaking dormancy in seeds with morphophysiological dormancy, which may act in isolation or in combination to induce germination in seeds. These include number first warm stratification, number second cold stratification. Warm stratification is sufficient to induce germination in some species, while in others the cold stratification induces germination. In some other plant species, warm followed by cold stratification induces germination, while in still others, cold stratification followed by warm stratification then again followed by cold stratification may be required to break the dormancy. Growth and dormancy break may occur simultaneously in some plant species, while in others, growth may be preceded by a dormancy break. Next type of dormancy is physical dormancy. Physical dormancy arises due to impermeability of seed coat or fruit wall to water. Physical dormancy has been reported for at least 15 different angiosperm families. It also occurs in several members of the families like Bixaceae, Melastomataceae and Winteraceae. With the exception of Canaceae, Musaceae and Nilambonaceae, all the plant families with physical dormancy in the seeds have large embryos and most of the food is stored in the embryo rather than in cotyledons. The seeds with physical dormancy have one or more lignified, that is cyclorid palisade layers in them. In addition to the development of impermeable layers in the seed coats or fruit walls, all the natural openings in the seed including micropyle, hilum and a callosal area also become impermeable to water. Hilum bridgin in legume seeds develops an additional layer of macrocyclarids called as the counterpalisade. However, an opening or passage through the palisade or other impermeable layers must be formed to make entry of water into the seed possible at the time of germination. In Papilionaceae and Mimosoideae, water enters seeds through strophiole, while in Canaceae, water enters through imbibition lid. Impermeable seed coat is not a family trait in plants, and many plant families have some members with impermeable and others with permeable seed coats. In Anacardiaceae, seed coat is not well differentiated and fruit wall protects the embryo. Dormancy in some members of this family arises due to impermeability of endocarp to water. Next type of dormancy is physical plus physiological dormancy. Seeds with impermeable seed coat mostly have non-dormant embryos. However, in some species, impermeable seed coats occur in addition to dormant embryos. The presence of physical and physiological dormancy in a seed is known as a combined dormancy, and germination requires overcoming both the kinds. In some cases of a combined dormancy, physical dormancy is broken before physiological dormancy, while in the others, reverse is true. Next type is the chemical dormancy. Some seeds fail to germinate because of presence of germination inhibiting substance in the pericarp. Such dormancy is known as a chemical dormancy. In addition to pericarp, germination inhibitors occur in embryo, endosperm, and seed coat, and in some species in structures that are dispersed with the seeds. The inhibitors include coumarins, para-ascorbic acid, ammonia, ferulic acid, and abscisic acid, etc. The inhibitors that block embryo growth are either synthesized in the seed or are transported to it from elsewhere. Most of the seeds which express a chemical dormancy also have physiological dormancy. For a seed to germinate, leaching of these inhibitors or their sequestration is required. Usually, at the time of germination, the ratio of germination inhibitors and germination promoters is tilted towards germination promoters. Some of the germination inhibitors are water soluble and are easily removed from the seed when it is exposed to sufficient quantity of water, and other seed inhibitors require a metabolic intervention for their removal. In some plant species, light or temperature may be involved in removing germination inhibitors from the inner parts of the seed like embryonic axes. Now the mechanical dormancy. Dormancy that arises due to hard and woody fruit wall is known as a mechanical dormancy. Usually endocarp forms the hard layer which inhibits germination in most of the cases, but in many species mesocarp is woody. Stony endocarps are found in some or all members of families including Anacardiaceae, Apocynaceae, Burseraceae, Cornaceae, Aliganaceae, Eliocarpaceae, Juglandaceae, and Lecithidaceae, etc. In the Anacardiaceae, Rhamnaceae, and Teliaceae, the endocarp can be impermeable to water, resulting in physical dormancy of seeds. In other families, the endocarp is permeable to water, but germination does not occur until fruits receive dormancy breaking treatment. Some fruits with stony endocarp have been reported to have embryos with deep physiological dormancy. Consequently, these require long periods of cold stratification to break dormancy. The fruits in tropical families may require warm stratification, but this has not been documented. In Oleaceae, Rosaceae, and Juglandaceae, the endocarp splits into two equal halves, but in many other families, only a part of endocarp is pushed aside by the emerging radicle like opening of a lid. Apart from the above classification, dormancy is also categorized into innate dormancy and enforced dormancy, depending on whether it results from environmental factors or from the factors internal to seed. Dormancy due to external factors is also known as imposed dormancy or quiescence. Overall, different kinds of seed dormancy can be attributed to one or more of the following factors, including number first, hard seed coat, number second, seed coat impermeable to water, number third, seed coat impermeable to gases, number next, rudimentary embryos, number 5, dormant embryos, number 6, germination inhibitors, and number 7, hard pericarp. Now we'll move on to seed germination. Germination is the process of coming out of seedling from the seed. Germination occurs only when seed is not dormant and all the conditions for its growth, external as well as internal, are satisfied. Under proper conditions of moisture, water, oxygen and temperature, seeds resume physiological functions and become active. The radicle protrudes out from the micropyle and pilumule also grows. Water is the primary requirement for germination. Oxygen is also essential, though germination can proceed for some time even in the absence of oxygen. Optimum temperature is essential factor in seed germination. Light is required only in case of positively photoblastic seeds. Seedlings grown in dark are frail, lean, and yellow. They are described as etiolated. Among the internal factors for germination, viability of seed is the most important one. Completion of dormancy is essential before germination can take place. Another requirement is that for food, food is stored in the cotyledons, endosperm, and in some cases in perisperm. Now we'll talk about these factors one by one. First, water. Seeds must attain some minimum species specific moisture content before they germinate, as they lose considerable amounts of water during maturation. For example, orthodox seeds lose up to 30% of their initial moisture content, and the recalcitrant ones lose considerably lower amounts of water during their maturation, which is 5% of their initial moisture content. Imbibition of water by seeds softens the seed coat, which helps break it open. Water also activates hydrolytic enzymes, which metabolize stored food materials. Both too low and too high amounts of water inhibit germination. However, the optimum value varies in different species. For example, seeds of Typha latifolia germinate better underwater than on a moist filter paper. Pre-soaking in water before sowing stimulates germination in caryopsis of Scolocloa festucacea. Next factor is temperature. Temperature has a profound influence on rate, potential, and onset of germination. Different temperature regimes may moderate the effect of water on seed germination. There is a specific soil temperature for each plant species at which the germination percentage is highest and time of emergence from the seed is lowest. As temperature moves on either side of this optimum scale, germination as well as time to emergence is affected negatively. Thus, thermal limit for germination is defined by minimum, optimum, and maximum temperatures, which vary for different species, and also control geographical distribution of a species. The minimum temperature for cucurbits is 15 degrees Celsius and maximum is 38 degrees Celsius, while as optimum temperature for germination ranges between 20 to 32 degrees Celsius. The optimum temperature for germination in most of the plant species lies between 16 to 24 degrees Celsius. Temperature works in unison with light to affect germination in many species. Next factor influencing seed germination is light. Germination in many seeds is influenced by light. Such seeds are known as photoblastic seeds. Photoblastic seeds can be positively or negatively photoblastic depending on whether light inhibits or promotes germination. Positively photoblastic seeds do not germinate unless exposed to a brief flash of light. Seed germination in grasses, Lectuca stiva, Senecio madagascariensis, Verbascum thapsus, Rumex crispus, Lathrum salicaria, and Physalis alkikangi, etc., is positively influenced by exposure to light. On the other hand, seed germination in some plants, for example, Citrulus lanatus, is inhibited by red, far red, blue, green, as well as white lights. Seed germination in some plants is not influenced by light. For example, seeds of Ceratocarpus arenarius, Calotropis procera, and Leptadenia pyrotechnica. Seeds, both positively and negatively photoblastic, respond to the quality of light to which they are exposed at the last. Next factor influencing seed germination is oxygen. Oxygen is required in aerobic metabolism of food products stored in the seed. Energy that is released during respiration of stored food materials forms the main source of energy for cell division and elongation of cells during seed germination. Though germination can proceed for some time in absence of oxygen, however, complete and healthy growth of seedlings is not possible in absence of oxygen. Next factor influencing seed germination is pH. Seeds of many plant species germinate at a wide range of soil pH. Many others, however, have a specific pH requirement. For example, the optimum pH for germination of Calluna vulgaris is 4, but it's 7 for Byrines bitrinata. In water sand system for seed germination, pH is usually maintained between 6.0 and 7.5, as even some calcifuge species like Alopicurus, Festuca, and Lolium, etc., germinate well at 6.5. Now we'll talk about types of seed germination. Number first is the apigeal germination. When the hypocotyl elongates quickly, bringing the cotyledons in the air, the germination is said to be apigeal. Apigeal germination is seen in many dicots, for example, castor, tamarind, gourds, and rarely in monocots, for example, onion. Number second type is the hypogeal germination. When the epicotyle elongates quickly, leaving the cotyledons on the ground or inside soil, the germination is said to be hypogeal. Hypogeal germination is seen in many dicots like pea, gram, and most of the monocots. With this account of seed germination and seed dormancy, I will conclude the present lecture and say goodbye to you.